Good afternoon to all of you. And today we are back again with the topic of enzyme inhibitions. Let us understand what are the enzyme inhibitors, because in some previous of my lectures we were dealing, which is called as the basics of enzymes. Then we went along with classification of enzymes, types of enzymes. We have seen there are examples. Now let us come to a part of enzyme inhibitions. Now, in a very basic sense, we'll quickly revise what we have seen. That enzyme is substrate that increases the rate of reaction. Any sort of reactions, if we want to increase the rate in the sense what? Increase its capacity or increase its ability to degrade that particular product, then the enzymes are said to be playing a very important role. So reactant binds to the enzymes and the product are released, and the enzymes can accelerate the reaction as much as 10⁻⁴ overall uncatalyzed reactions. म्हणजेच याचा अर्थच असा होतो की समजा एक आपल्याकडे जर एक constituent आहे, let us suppose if A is there and this A has to be converted into B, and in a normal sense if we do not put any sort of enzyme to it for its own conversion from A to B for catalyzing this reaction by itself, it is very difficult for that particular constituents. मग याच्यासाठी काय केलं जातं, तर याच्यासाठी त्या रिएक्शनला एनहान्स करायसाठी that A gets converted to B, we put an enzyme in front of it, and because of enzyme the reaction increases 10⁻⁴ times. 10⁻⁴ times ची रिएक्शन इतक्या पटकनशी वाढली जाते, and because of that the catalysis reaction would take place and the substrate would be degraded into product B in a very quick sense and a very forwarded sense. Specificity of enzyme towards the substrate is controlled by its structure. Of course, जेव्हा active site of an enzyme चा जेव्हा part आपण बघत होतो, at that time we have already seen this part that specificity towards the substrate, it has been controlled basically with the help of the enzyme. की कोणत्या प्रकारचा सबस्ट्रेट कसा स्पेसिफिकली बाइंड होणार, आपण बघितलं होतं ऍक्टिव्ह साइट मध्ये की तिथे असणारे जे अमिनो ऍसिडचा जो सिक्वेन्स आहे, that sequence is said to be one of the important sequence wherein the molecules which come along with the substrate have certain amount of interactions, and these interactions will be helpful for binding of the substrate to the enzyme and degradation of that substrate into product P. Okay, this unique fit of substrate with the enzyme controls the selectivity of substrate for its product formation. हे आपण मागे बघितलं होतं की enzymes are said to be very specific towards the substrate, as well as there are certain substrates which only act or react with the enzyme which are very much highly specific. So specificity of the enzyme reactions would be there, and the reaction would take place only if the binding is said to be very proper in a very correct manner. Then and only then the reaction would be forwarded. Otherwise, if the binding is not in a proper sense, then the substrate would leave itself from the enzyme, and the intact enzyme will be, or the hollow enzyme will always be like that. So substrate specificity plays a very important role. Now, what is enzyme inhibition? Inhibitors are substances that bind to an enzyme and interfere its activity. आता मागच्या पण एक लेक्चर मध्ये कालच्या दिवशी एक मेकॅनिझम बघितलं होतं, which is called as feedback mechanism. की फीडबॅक मेजरमेंट मध्ये अशी एक फिगर आपण काढलेली होती, आणि त्याच्यामध्ये मेन्शन होतं that the product A gets converted to B, B to converted to C, C is the final product, and once the final product has been generated, this final product will come and bind to the first enzyme of the pathway and inhibits it. मग हे कशामुळे होतं, तर ते इनहिबिटर्स मुळे होतं. कशामुळे? binds to its enzyme and interfere its activity. हे एन्झाइमलाच बाइंड आधी करून टाकतात, जेणेकरून the substrate which is present over there will not be able to bind to that enzyme. The enzyme ला ती binding properly होत नाही, and therefore the reaction cannot be there. Therefore the inhibitor plays a very vital role in blocking the enzyme, or it doesn't allow the matching of that particular substrate to the enzyme. It can prevent formation of enzyme substrate complex, or can prevent ES breakdown to enzyme and product. बघा, म्हणजे नुसते एन्झाईमला बाइंड नाही होऊ शकत. एन्झाईम सबस्ट्रेट बाइंडिंग असलेला ES कॉम्प्लेक्स जरी डेव्हलप झालं, स्टिल अगेन the particular inhibitor would go and bind to an enzyme substrate complex, and it will help, or it will remove that enzyme substrate complex where the free enzyme will be there and the substrate will be left out. Inhibitors are chemicals that reduces the rate of enzymatic reaction. आता प्रश्न असा निर्माण होतो की एकीकडे आपण बघतोय की सेलच्या आतमध्ये there are certain amount of enzymes or certain amount of reactions that are taking place, and then on the other side we are saying what is the use of these inhibitors. मला सांगा प्रत्येक वेळेस जेव्हा एन्झाईम वर्कआउट करताय विथ द हेल्प ऑफ व्हॉट इट इज वर्किंग आउट विथ द हेल्प ऑफ एनर्जी, आणि तेवढी एनर्जीची गरज जर नसेल तर विनाकारण सेलनी एनर्जी घालवायची गरज नाही. Therefore, in every sense it will be that these inhibitors will help to reduce the rate of reaction. पटकन जशी ती रिएक्शन व्हायची त्याला एटीपी फॉर्मेशन एक्सेस अमाउंट मध्ये लागणार आहे अशी कंडिशन तिथे डेव्हलप न हो, किंवा if the reaction of if the product that is being formed is at an optimum level, अशा वेळेस ऑप्टिमम लेवलच्या पुढच्या गोष्टी आपल्याला करायची गरज नाहीये. अशा वेळेस द रेट ऑफ रिएक्शन हॅज टू बी स्लोअर डाऊन, अँड व्हॉट हेल्प्स इन दॅट, द इनहिबिटर्स ऍक्च्युअली हेल्प्स इन दॅट. Enzyme plus substrate, ES complex, enzyme plus product. This is a very basic reaction that we have seen from my first lecture as well. That generation of ES complex would take place, binding of enzyme to the substrate and formation of complex would take place. Then moving further, the enzyme will be there and the product P will always be released. Now, enzyme plus substrate plus inhibitor. आता inhibitor इथे टाकल्यावरती काय होतं बघा. If it forms a complex called as enzyme substrate inhibitor or enzyme inhibitor. म्हणजे इथं लक्षात घेण्यासारखी आपली गोष्ट आहे की इथं बघा, इथं काय आहे, इट इज जस्ट an ES complex. Once ES complex formation takes place, this reaction will move forward and the substrate here it will be degraded or converted into terms of product P. Enzyme बदलला का? नाही. Enzyme remains as it is, but this substrate is now converted into product P and product generation would take place. But when there is an inhibitor, जेव्हा inhibitor त्या reaction मध्ये आहे, at that time, see this, enzyme substrate and the inhibitor is there, and the inhibitor may have direct contact with the enzyme. या एन्झाईमला जाऊन इनहिबिटर अशा प्रकारे लागणार आहे की जेणेकरून अल्टिमेटली देयर विल बी नो प्रोडक्ट फॉर्मेशन. व्हाय? बिकॉज this inhibitor will block all the sites in the enzyme for binding of the substrate and development of enzyme substrate complex. If these things doesn't work out in a proper way, then there will be no product formation that would take place. There can be irreversible or reversible inhibitors. दोन प्रकार आहेत, रिव्हर्सिबल इनहिबिटर्स आणि इररिव्हर्सिबल इनहिबिटर्स. रिव्हर्सिबल इनहिबिटर्स काय असणार आहेत की जे काही काळापुरता the reaction they will stop that reaction for some period of time. So once there is always a need of ATP or need of that particular product, they will release itself. Now the substrate will bind and product formation or ES complex will take place and the reaction will be conducted. But there are certain inhibitors which are irreversible inhibitors की ज्याच्यामध्ये ते inhibitors reverse येऊ शकत नाही. So once they bind to that enzyme, they will never allow the substrate to bind to that enzyme and no product formation would take place in that reaction as well. See this. Irreversible bind to the enzyme through covalent bond. Reversible bind to the enzyme through non-covalent interactions. Irreversible bind to covalent bond, एकदा covalent bond ची binding झाली की they will not reverse itself, they will bind directly to the enzyme and will never allow the substrate to again bind to that particular enzyme. ही प्युअरली कोव्हॅलंट इंटरॅक्शन असणार आहे, एकदम स्ट्रॉंग बॉन्ड डेव्हलप होतो and therefore either the enzyme will be lost and the substrate will not be degraded into product P. While in case of reversible inhibition, it may be non-covalent interactions or it may be certain kind of hydrogen linking that would take place. So that after some period of time, the inhibitor will move out from the enzyme and this time the substrate which is specific for that particular enzyme will bind to that enzyme and formation of enzyme substrate complex will take place. Generation of product P will take place. Enzyme inhibition. आता महत्त्वाचं. Reversible enzyme inhibitors. Inhibition of enzyme activity in which the inhibitor molecule entity can be associated or dissociated from the protein's binding site. Reversible आहे. हे सांगितलं तुम्हाला. Reversible असल्यामुळे काय होणार आहे की तिथे त्या प्रोटीन बाइंडिंग साइट म्हणजे जी ऍक्टिव्ह साइट आहे त्याला जाऊन बाइंड होणार and it can also move out from that particular site and binding of substrate would also take place. Three types of reversible inhibitors. कोणकोणचे? Competitive, uncompetitive, non-competitive. लक्षात घ्यायचं विद्यार्थी मित्रांनो. आपल्याला याच्यामध्ये एक्झाममध्ये पण हा प्रश्न विचारला जाऊ शकतो. व्हॉट आर द डिफरंट टाइप्स ऑफ इनिबिटर्स दॅट आर प्रेझेंट. 3 टाइप्स ऑफ इनिबिटर्स, कॉम्पिटिटिव्ह, अनकॉम्पिटिटिव्ह अँड नॉन कॉम्पिटिटिव्ह इनिबिटर्स. वन बाय वन वी ट्राय टू स्टडी. सगळ्यात पहिला कॉम्पिटिटिव्ह इनिबिटर or competitive inhibition. These compete with the substrate molecules for the active site. The inhibitor's action is proportional to its concentration. It can reverse inhibition by adding more substrate. आता इनिबिशन म्हणजे याच्यामध्ये काय बघा, लक्षात घ्यायची गोष्ट आहे की इट इज अ कॉम्पिटिटिव्ह इनिबिटर. असं म्हटलेलं आहे कॉम्पिटिटिव्ह इनिबिटर. मग तो काय करणार आहे, इट इज हॅव्हिंग कॉम्पिटिशन. कोणाशी कॉम्पिटिशन आहे इनिबिटरची? कॉम्पिटिशन विथ द सबस्ट्रेट. सबस्ट्रेट आणि इनिबिटर दोन्ही सेम सारखेच आहे. आणि व्हॉट दे आर फायटिंग और व्हॉट दे आर कॉम्पिटिटिंग फॉर बाइंडिंग ऑफ देमसेल्व्स टू दॅट पर्टिक्युलर काइंड ऑफ एन्झाइम. त्या एन्झाइमला बाइंडिंग साठी जो पहिले बाइंड होईल इट विल नेव्हर अलाउ द अदर पर्सन्स और अदर थिंग्स टू बाइंड. म्हणजे फॉर एक्झाम्पल एक एन्झाइम आहे, त्या एन्झाइमला दोन्ही कॉम्पिटिटर्स आलेले आहेत आणि अशा वेळेस इफ द सबस्ट्रेट बाइंड्स डायरेक्टली द इनिबिटर विल नॉट बी एबल टू बाइंड. और इफ द इनिबिटर बाइंड्स डायरेक्टली the substrate would not be able to bind. इनिबिटर आला, इनिबिटरचे हे बघा तुम्ही मॉर्फोलॉजी बघा कशी आहे. and this is the active site of an enzyme, and once the inhibitors goes and bind to it will never allow the substrate to it. आणि हे कॉम्पिटिशन आहे एकमेकांमध्ये. these are competitive and because of this competition itself it will they directly bind to the enzyme. It will never allow the substrate to bind to that particular enzyme and the ES complex will not take place. Product formation is stopped. Malonate is a competitive inhibitor for succinate dehydrogenase. लक्षात घ्या. Malonate स्ट्रक्चर बघा, CH2OH, CH2COH. सेम succinate dehydrogenase नावाचं एन्झाइम आहे. याला बाइंड केल्यानंतर इट विल हॅव अ कॉम्पिटिटिव्ह इनिबिशन. सक्सिनेट टू फ्युमारेट रिएक्शन ब्लॉक करण्याचं काम मॅलोनेट करू शकतो. दिस इज वन ऑफ द बेस्ट एक्झाम्पल्स ऑफ competitive inhibition. Uncompetitive inhibitions. Inhibitor binds to the enzyme substrate complex. Binding site for inhibitor is created only upon substrate binding. There is no EI complex, only E, ES and ESI, but ESI can't make product. Tetramethylene sulfoxide and 3-butylyl 1 oxides are uncompetitive inhibitors of aldehydes. आता याच्यामध्ये गंमत अशी बघा, अनकॉम्पिटिटिव्ह मध्ये नॉर्मली इन अ नॉर्मल सेन्स एन्झाइम प्लस सबस्ट्रेट गिव्स एन्झाइम सबस्ट्रेट कॉम्प्लेक्स and then it will give enzyme plus product P. Okay, fine. See this substrate binding here and binding after binding this is the ES complex that is been developed. आता या ES complex ला कोण बाइंड होतोय? here ES complex ला आता ही रिएक्शन जर फॉरवर्ड झाली तर अल्टिमेटली प्रोडक्ट P जनरेशन होईल. Now for this purpose, when ES enzyme substrate and the inhibitor will binds, it binds to enzyme substrate and inhibitor. See this. ES complex ला inhibitor binds झाला and it modifies the active site of an enzyme so that this particular the substrate will not be degraded and product formation will not take place. हे लक्षात घ्यायची इथं गोष्ट आहे. सगळ्यात महत्त्वाचा पॉइंट इथं त्यांनी आपल्याला सांगितला तो म्हणजे हा. There is no EI complex, enzyme inhibitor complex डेव्हलप होत नाही. त्या पहिल्या ज्या होत्या तेव्हा एन्झाइम इनिबिटर कॉम्प्लेक्स डेव्हलप होऊ शकतो, पण इथे नाही होणार. इथे काय होणार आहे, एक तर ES और ESI. ESI म्हणजे काय, एन्झाइम सबस्ट्रेट इनिबिटर कॉम्प्लेक्स डेव्हलप होईल and it will never allow that substrate to get degraded into product P. So it will be, there will be a metabolic block or halt will be there and ultimately the product will not be taking place. Non-competitive inhibitions. These can bind to enzymes and enzyme substrate complex. Enzymes can bind both substrate and inhibitors, but ESI complex can't make a product. Inhibits by binding irreversibly to enzyme, but not at the active site. Active site ला सोडून the binding होऊ शकते. For example, cyanide combines with iron in the enzyme cytochrome oxidase. Non-competitive inhibition मध्ये बघा. Non-competitive inhibition changes shape of the enzyme so that it cannot bind to the substrate. Non-competitive. काही कॉम्पिट करायचं नाही, कोणाशी काही कॉम्पिट करायचं नाहीये, but it will bind to the enzyme. आणि enzyme bind झाल्यानंतर ultimately it modifies the active site. Active siteच modification. In case of, एक गोष्ट लक्षात घेण्यासारखी आहे, that in case of non-competitive inhibition, the inhibitor will never bind to the active site of enzyme. Instead, it will bind to some other site in the enzyme and it try to modify the active site of an enzyme. ही पाहिजे ही active site. आता काय व्हायला पाहिजे होतं नॉर्मल सेन्स मध्ये हा सबस्ट्रेट इथे बाइंड व्हायला पाहिजे होता, पण विथ सिन्स कॉम्पिटिटिव्ह एक्झिबिटर इंटरफेस विथ ऍक्टिव्ह साइट ऑफ द एन्झाइम सो सबस्ट्रेट कॅनॉट बाइंड. हे मॉडिफिकेशन केलं अशा रीत्या मॉडिफाय झाला आणि therefore substrate cannot bind to this particular thing. This is called as uncompetitive inhibition. Competition दोघांमध्ये नाहीये, but it will directly bind to the enzyme and after binding to the enzyme gets modified in such a kind of way that it will it will have some problems with the substrate. Irreversible enzyme inhibitors. The binding of inhibitors can stop a substrate for entering the enzyme active site for catalyzing its reaction. Irreversible inhibitors usually react with the enzymes and change its chemical. They bind covalently and destroy the functional group of the enzyme that is essential for its activity. Competitive and allosteric. आता हे irreversible आहे. Direct competitive inhibitor म्हणून तिथे टाकायचा and it will destroy that enzyme so that the substrate will never bind to that particular active site. And this is again allosteric, which is another part. We'll study this allosteric site very coming of my lectures because this is one of the most interesting topic in this particular aspect as well. So this was very short part that we have covered up today, but it will certainly have a more amount of weightage that is developed in your paper as well. Thank you for